My name is Scott Bell, and on behalf of TRI, I'd like to welcome everyone to the NCRIS Capabilities Seminar this afternoon. We're delighted to see so many of you in person, and I know that there's an equal number online. I'd also like to acknowledge the traditional custodians of the land that we are on today and recognise their continuing connection with land, waters, and community. We pay our deep respect to them and their cultures and to elders past and present. So shortly after arriving at TRI 4 years ago, my then colleague, Graham Galloway, mentioned the term NCRIS. And I said to Graham, "What the heck is NCRIS?" Since then, you'll be reassured to know that I've learnt quite a lot about NCRIS, but I do suspect many similar biomedical researchers based at TRI are in the same boat that I was. Limited knowledge about NCRIS, especially in what the facilities can offer to my team. Today we're here to learn about the extensive NCRIS facilities and services that aim to accelerate therapeutic discoveries, foster collaboration, and facilitate research translation. NCRIS is the National Collaborative Research Infrastructure Strategy. Now, I still don't really know that without my notes. Funded by the Commonwealth Government and aims to ensure researchers have access to cutting-edge research infrastructure, everything from assisting with small molecule manufacturing to access to massive microscopes. TRI is building an ecosystem where we welcome researchers, industry, clinicians, and support services, including NCRIS, to collaborate and accelerate research. In that ecosystem, TM at TRI is the final piece of the puzzle that will support the translational pipeline of biomedical researchers to allow scale-up manufacturing of early-stage biotechs in Queensland. Now I'll get out of the way and hand over proceedings to the event MC, Professor Paul Bonington from the University of Queensland, who will be your facilitator this afternoon. Welcome, Paul.
Thank you, Scott. And so good afternoon, and thank you for taking the time to attend our NCRIS Capabilities Seminar. And it's the first of many, I hope. We hope this format gets repeated around Australia as the year progresses. And the intent here is to better connect and inform researchers of the amazing national scale research infrastructure we have in Australia. So I'm Paul Bonington, but the local folks here call me Bono from UQ, Pro Vice Chancellor for Research Infrastructure. And I've been at the university now 18 months. I arrived from Monash University. So it gives me great pleasure to welcome you all here today and to be your MC for this event. This is our very first event for 2024. We have an amazing lineup of speakers. They are representatives of the NCRIS research facilities, which support therapeutic discoveries, not just here in Queensland, but right across Australia. And we have a great turnout, both in the room here, but also online, as Scott has mentioned. There are, of course, a large number of registered attendees, and I think that's a testament to the interest here and the critical capabilities that we're going to highlight today. You'll see how the advanced capabilities and infrastructure can enable progress therapeutic discovery across every stage of the R&D life cycle. Now, of course, therapeutic discovery is a long and complex process, and we're very fortunate here in Australia that we've got the federally funded facilities under the, as Scott has mentioned, the acronym NCRIS, National Collaboration Research Infrastructure Strategy, here to assist you as researchers. Now, I'd like to say that the most important letter in NCRIS is C for collaborative. All of the facilities are established and operated as partnerships. Along with the federal government investments, they are co-invested by universities and institutes like this, critical capabilities in the research sector, and also by local state governments. The facilities come about because no single organisation can do this alone. Building research world-class research infrastructure is challenging. Partnerships and collaboration are critical to making this happen. Secondly, as our Vice-Chancellor Peter Hoy used to say, cutting-edge research requires cutting-edge infrastructure. And I've been working in research infrastructure for a while, and I can attest to the importance of having the latest cutting-edge technologies and capabilities available to drive research and to be relevant to industry needs. It really does make a difference to the efficiency and the speed by which we discover, innovate, create, impact, and improve the human condition. And a great example of that is in the very building we are today. This building houses the Fraser Institute, named after Professor Ian Fraser AC, and he was a leading team member in the discovery of the HPV vaccine, which was successfully commercialised and rolled out globally in 2007. Now, Professor Fraser utilised numerous advanced research capabilities at the time, like flow cytometry, histology, recombinant protein expression, and purification of the vaccine to eventually get to this therapeutic discovery and get it from the research bench and into the health centres around the world. And more recently in 2020, during the 2020 pandemic response, Australia's vaccine candidate and the team behind the UQ molecular clamp technology utilised cryo-electron scanning microscope capabilities, high performance computing, cell line development, recombinant protein production, high-end assay and characterisation, and animal safety and toxicity testing, and all available here in Queensland. And of course, while collaborating with key facilities around Australia to progress work. These examples set the scene for what we'll discuss today. We're going to hear from 8 NCRIS facilities and delve deeper into the specific capabilities and how they can assist you along with every step of the way. So just to go a little deeper into what NCRIS is. It is a set of national scale world-class, oh, I've got a slide for this, here we go. A set of national scale world-class research facilities hosted across Australia, which offer subsidised access to researchers regardless of which university, institute or company they're from. And most of the facilities have been around for at least 16 years. The facilities have a very long history of enabling excellence in research. All of the facilities came about through a consultative, facilitated co-design process across the research sector, led by research and industry leaders. NCRIS is a systematic enabler of Australia's innovation system and is considered a national asset. I might say the envy of the world. And in fact, I believe NCRIS is probably the most important thing we do as a nation, but I am very biased. Now, the capabilities of these facilities are seamless and diverse. These capabilities include, for example, characterization like high-resolution imaging and microscopy, high-performance computing, e-research, advanced data services, fabrication, bioproduction, sequencing and omics, and many, many more. And these facilities are all at the forefront of technological innovation and research, and they have world-class expertise and instrumentation co-located, which can really help drive your research to another level. All of the facilities are supported by leaders in their field, and my PVCRI colleagues across the university sector always look to encourage research, industry and government partners to work with them. These experts are well placed to advise and guide the best practice when it comes to applying advanced capabilities and the latest technology to support research. So, without further ado, we'll move on to the presenters. Just a little bit of housekeeping. We will have time allocated at the end for questions and answer, a bit of a panel session, if you like. Do ask that those who are Zoomers, if you can use the Q&A function of Zoom and not the chat in Zoom when you want to ask your questions. And please feel free to bring those up at any time. But we'd ask the locals here to also hold your questions until the panel discussion. So with that, we'll move on now to our first speaker, and that is Dr. Stuart Newman, the CEO of Therapeutic Innovation Australia.
Thanks very much, Bono, and thanks very much, everybody, for taking the time. Now, I'm going to start in the middle, like those films that start in the middle and have a flashback, because I want to put this slide up and kind of talk a little bit about the process of translation and where TIA puts ourselves in that process. Now, we look at the world through a TRL sequence, whereas lower technology readiness levels are discovery, research, and very early stage, going through to development of potential therapeutics, putting them through to the market later on. Now, it's a long, very difficult process, but when it comes off, you can literally change the world. And we've seen that this last couple of years. But what I wanted to get from this diagram is that TIA, Therapeutic Innovation Australia, which is my organisation, we place ourselves right in the valley of death, right in between TRL's level 3 to 6, the support and expertise and capability gap that is observed between, which makes it very challenging to take a research discovery and translate it through to a product or a service with real world social, economic or health impact. So with that in mind, briefly introduce TIA. We are a network of research infrastructure facilities that are arranged around product types. So we support access to our facilities through investment in people and equipment at a network of key facilities around the country. We also have a voucher scheme, which I'll talk about a little bit later on. And we have an expert engagement team to make sure that researchers and SMEs know which facility to go to immediately and can save a lot of time in doing so. This is about the most technical slide, my entire presentation. It breaks down the kind of materials that TIA facilities can produce and help you develop, and how and the services associated with them. Start at the top. We have a very pretty comprehensive and integrated small molecule pharmaceutical pipeline, starting at compound storage and logistics at Compounds Australia, to local facility, going all the way through the process, through screening, medicinal chemistry, lead optimization, through to preclinical efficacy testing. Our second and a very new group, so new it's still got the plastic on it, is our RNA products capability. That's a network of 4 nodes, which have expertise in the design and production of high quality, lab scale, lab quantity RNA for research purposes. We have nodes in Brisbane, Sydney, Melbourne, and Perth. Our biologics and vaccines capability specializes in antibodies and proteins and other macromolecules through the discovery and engineering, production, and some process development as well. And finally, our cell and gene therapy capability is a network of highly advanced patient-facing GMP cell and gene therapy production facilities. And our materials range all the way from the small molecules, all the way through getting bigger and bigger in molecular weight, all the way through to cells. So this is where we are, and it's totally fair enough if your eye wanders up to the top right corner where all the Queensland based facilities are. But I just want to make the point that all these facilities around the country with their diverse and unique skill sets are available to Queensland researchers. Good case in point, we've had quite a lot of interest from Queensland researchers in the Monash fragment platform, for example. So all these facilities are available to you via TIA. Now, that was a lot of facilities. So how do you find them? Well, you ask these people. We have an engagement team, 3 individuals. Ben is here in the audience. He's based in, put your hand up, there we go. That guy there. He's based at University of Queensland, takes care of our biologics and RNA group. We have a Heather in Sydney, takes care of cell and gene therapies, and John in Melbourne takes care of small molecule therapeutics. These are the people who I will ask which facility a project should go to. So they're here to help you. They're intelligent signposts, very intelligent signposts, and they will get you to the facility and the expertise that you need. So it's just a few snapshots of the kind of thing that we support. My perennial favorite is the first one up here. We're supporting Peter Timms in developing a vaccine to help koalas fight off chlamydia. I always find it very impressive. An animal that sleeps 20 hours a day has time to catch anything, let alone chlamydia. But there's a real sort of variety in the kind of projects we can assist. Okay, last slide. We have an additional scheme which opens on March the 4th. It's our Pipeline Accelerator scheme. It's a voucher scheme that people can apply for to get some additional subsidy to access a TIA facility. It's a great way to get some proof of concept data for a grant or generate some material for further testing. It's open to SMEs and academics, and we can fund up to we can fund half of a quote to access one of our facilities up to a maximum of $50,000. Don't lose on the IP. It's entirely enabling. 55% success rate. You won't get much better than that in the current climate. So please come and see us either here at TRI, where we have an office on the 7th floor, or via our info at. We're here to help, and we'll be delighted to hear about that project and how we can advance it. Thank you very much, and talking to speaking to you after the event.
Okay, thank you, Stuart. Our next speaker is Jane Fitzpatrick, the CEO of the Australian National Fabrication Facility.
Thanks, Bono. I'm not as tall as Stuart, so I may wander over there at some stage. Lovely to be here. Thank you very much for the invitation and the opportunity to speak to you about what we do and how on earth it is anyway relevant to anybody who's doing medical research. We're the fabrication facility, so we work in the hard end of the world. So things actually making the physical objects and devices that work within the medical and the medical technology space. As everybody said, we're 17 years old. We work on a collaborative model. There are all the universities which host a facility for us. And again, we're dotted all around the country, just like our cohort here in NCRIS. So what is fabrication? To put it very simply, fabrication involves starting with a flat surface and then taking bits off or adding bits on. You build up layers, you build up functionality, and then you dice it all up into tiny little things and put them into a package. That's fabrication 101. If you want 102, you'll have to talk to one of my engineers. So this is what we do. To do all of those steps, however, you need an awful lot of tooling. We've got about 500 different pieces of tooling around the country covering all of those different areas. We also have over 130 expert staff, and that's one of the key things. You don't have to come in and spend 6 to 12 months learning how to use one of these pieces of equipment. You actually come in, you sit with an engineer, they walk you through the process of learning how to use that instrument, and within a week, you're getting real product of those instruments. So it's a really quick and efficient way of learning how to use these techniques. We can train you, we can give you direct access to the instruments. We can also do fee-for-service work. So if it's a one-off, you don't need to spend the time learning how to do these techniques, that's fine, we'll do it for you. And some of our techniques are too onerous for most researchers. Some of them will take you a bit longer to get up to expert level. So if you need expert results, then you pay us to do it and we can do it for you. One of the things that we do an awful lot of is talking to people about their project and telling them they can't do it. A lot of people come in with a publication and say, hi, I'd like to do this. And we go, that's 24 different fabrication steps. It's going to take us a year to optimize that on our tools. Let's do something different, shall we? But that's the thing, we'll give you an option to do something different that will actually get you to the same point, hopefully, but in a shorter and much more resource light manner. A bit of funding there. One of the things that I'd like to highlight on the fact that I've got that money, those numbers up there. For every $1 you get charged for access to ANFF, somebody else is paying $4. And that's just the operational recovery. That doesn't include any of the CapEx that you are accessing. So for every dollar you spend, somebody else is paying for, and there's not many deals quite as good as that. This is just to notice these are the people. They are actually real. They do work for me. They all have a t-shirt. This is how I know they work for me. All right, so they are not researchers. They're not doing this part-time. They're not giving their time as a favor to your supervisor. They're dedicated professionals there to help you do what you need to do. Not everyone understands what fabrication is or how it can help. So we've put together an online learning portal to help people get the basics of the physics that's behind some of the fabrication technology that we use. This started during pandemic because we couldn't train people face-to-face for long periods of time. So we needed to find ways of giving them that background theory knowledge without having to doing face-to-face, because we had 1 hour training sessions. That's all we had. These courses are free. These courses are easily accessible through our website, and anyone can register. Anyone can do any of the courses. You get badges, and you can build up your own knowledge should you need it. It also ensures that you've got a base level of understanding of what you're doing before you come and talk to our engineers. It makes our engineers, the information our engineers and scientists can give to you, much more valuable. That's at the top of our website. That's where the button is. Just to give you an idea of the size of our facility, and this, remember, we're only one of 24 facilities. These are the stats from the FY23, so nearly a year old at this stage, but certainly indicative of what we do on an annual basis. Over 3,000 people, over 24 years of usage, 630 publications, which do better than GO8, I'd like to say, which is really nice because it means that we're supporting the really world-class research that we are here to do. That's what we're here tasked to do. You also see how many training instances there. So even though we're got 3,000 clients, we're also training over 3,500 different times on a yearly basis. Of 130 people, that's a lot of training. Very briefly, we have a case study book that's up on, and if I just, so that's our case study book. I've just pulled out some of the highlights from it that you might be interested in. One is a textile project. How do we recycle textiles here in Australia? It's a world leading capability on breaking down those fibers, bringing them back to reusable fibers and working that through. And that is why we're involved, because it is nanotechnology in that respect, and it's also fabrication. Optical probes, implantables, we do a whole range of them for everything from brain function all the way through to microfluidic drains in eyes. We talk about how people are working with hydrogels. How do you characterize those hydrogels? How do you use them? How do you deliver them? And also then working at that computer brain interface. How do we look at the brain, use that information, and then create solid state computing in silicon that mimics that? So you can see that even using the hard and inspired by the soft, but also using the soft and how to use the hard to deliver those capabilities. This is my last slide. So Stuart very kindly started the TRL conversation, so I will continue it. We work in general around the TRL 1 to kind of 6 mark. However, we also are aware that there is a valley of death. So we've put in a program, not unlike what Stuart has done at TIA, to try and help our cohort get through that valley. Ours is slightly different, slightly smaller in scale, in that we're trying to give them out of lab support. So we're not trying to get them onto our tools. We're trying to give them patent support or regulatory analysis, marketing analysis. What do they need to make their particular early stage entity more investable for people? So it's called AMP-C. and it has $20,000 and $100,000 grants that are non-dilutive funding to an early stage entity. And again, it's about providing them with the stuff you can't pay for out of grants. And that's the key. If you're an academic, you can't pay for a marketing analysis. You cannot get a manufacturing feasibility study done. We'll help pay for those kinds of things. And that's me. So thank you very much for listening.
Great. Thank you, Jane. Our next speaker is Sarah Richmond, who is the General Manager for the Science Program. Oops, there we go. Bioplatforms Australia.
Thanks, Bono, and thanks everyone for the opportunity to talk with you today. Bioplatforms is the NCRIS-enabled infrastructure provider for genomics, proteomics, metabolomics, and most recently in synthetic biology. And we support these infrastructures through investment in people as well as in instrumentation. And we support these capabilities through a national bioinformatics investment called the Australian Biocommons, which is really tasked with helping researchers take data off these instruments and machines and go and do really useful things with it, whether that be around data management, metadata, to data movement, to data analysis in environments that are optimized for biomolecular research data. We have around 15 facilities across the country, and hopefully some of the names on this list are familiar to many of you in the room. Last year, these facilities serviced around 20,000 research contracts or projects, and perhaps unsurprisingly, about half of those were from biomedical or diagnostic researchers, with the other half kind of split across agriculture and food and environment and biodiversity. What's perhaps most important about this slide is that although there's a bunch of facilities across the country and they all sit within their own institutions or universities and somewhat act a little independently, they do form part of a really large and connected collaborative network that have shared cross-disciplinary expertise. We routinely bring our facility members together to help us with knowledge sharing, sharing their experiences and solving big challenges. So we're able to kind of harness this national expertise to apply to your research projects. We then deploy all of this awesome expertise and capability into what we call national research priorities or national data initiatives. These are really focused around building large-scale referential data resources, which we see as critical underpinning infrastructure for research. And we do this into 3 prioritised research themes, again, around biodiversity, human health, and agriculture and food. Each of these hexagons you see on the screen end up receiving around $1 million investment from bioplatforms via NCRIS and get that plus more from co-investment from those communities. You heard Bono say it before, and I think everyone before me has mentioned the word collaboration. These national projects are really around communities of practice and using those communities to help collaborate and drive forward national agendas to deliver really large-scale referential data products. The beauty of doing it this way is that we're able to catalyse scientific collaboration onshore, enhancing our sovereign capabilities, but also enables us by kind of banding together and delivering these open access sort of public good data resources that are attuned to an impact now, but are future-proofed through fair data principles and so on. We're able to really accelerate the translation of research into impact and into industry. Our health and medicine theme aligns really tightly to MRFF and NHMRC, and we often partner with grant recipients across these programs. We've invested pretty significantly in some national initiatives that work on better understanding of cancers such as melanoma, and into supporting better health outcomes for Indigenous Australians through the National Collaborative Indigenous Genomics Initiative, and most recently into the Australian Alliance for Indigenous Genomics, or ALIGN. We've also recently been really interested and invested in working with a lot of our collaborators in that nexus of nutrition and human health and what that means for kind of healthy living. And we've been investing in some programs around, as an example, one called the Plant Protein Atlas Initiative, which is around doing some deep omic, multi-omic characterization for various pulse varieties in Australia to look at different protein compounds. Finally, I just wanted to touch on that we're actively supporting various junctions with our fellow NCRIS peers, who you'll all hear from today, around looking at programs and research projects that interact with a number of different research infrastructure requirements. I think increasingly we're finding that researchers are not just doing genomics or proteomics or imaging, they're interacting with various groups of our facilities at the same time or going from one facility to the other. And as a group, we're really wanting to make sure we can support researchers in that agenda, make it a bit more seamless, like you're interacting with one facility, even though you might be interacting with 8. And so if you've got any ideas around this, we'd be really, really keen to have a chat with you. Thank you.
Okay, thank you, Sarah. Our next speaker is Marina Trigueros, Communications and Outreach Coordinator from Phenomics Australia. Thank you.
Hello, good afternoon, everybody. So Phenomics Australia was established in 2007, and it's a founding member of NCRIS. Since then, we have expanded to 20 locations across Australia. And through these openly accessible nodes, we offer specialized infrastructure, research services, and technical expertise to drive the discovery of the molecular basis of disease to benefit the health of all Australians. My name is Marina Trigueros, and I'm the Communications and Outreach Coordinator for Phenomics Australia. Phenomics Australia team is formed from a group of board that is a group of experts that they do oversight the Phenomics Australia investments and activities, and they do provide strategic guidance. We also have a group of scientific directors that are experts and also the leaders of the different nodes that we have around Australia, and the Phenomics Australia executive team that is responsible of the day-to-day operation of Phenomics Australia. As I said before, we are in 20 locations across Australia. We have more than 100 projects per year and more than 100 million of investment per year. We have partnered with world-leading universities and research institutions, medical research institutions, where all our technical experts are based. We offer different services, but main ones are biobanking, in vivo and in vitro, genome engineering and disease modeling, genotyping and phenotyping, functional genomics, and high-throughput screening and pathology. But from all of those ones, we offer more than 38 specific research services. All of those ones are here. You can take a look on our website to check the different and specific research that we have. But also for the ones that are here, when we go out to the networking part, we can take the flyers that I brought with me. As you can see, the array of different expertise that we can offer is a quite comprehensive one. So how is this expertise being used? In this slide, I'm going to show you some of the examples that Phenomics Australia expertise is enabling here in Australia, but also around the world. For example, we have our histopathology team at the University of Melbourne has been working with Monash researchers to identify new antibiotic that can target the multi-drug resistant bacteria. Those ones just as a huge health problem globally. Also, I'm pretty sure you all have seen or you are aware of the Catherine Folbig case, where a genetic study done at the Australian National University and supported by our in vivo services discovered that the fulvic children had a rare genetic mutation that was provoking fatal cardiac arrhythmias. And these findings were played a major role in the case being checked again and the final pardon being handed out recently. Also, actually, quite recently, last week, one of our supported projects made the news because researchers at the NU were able to find a protein that is able to slow down the division of the bowel cancer cells. And this is quite important, not only for being able to treat the bowel cancer, but also to detect it in tests quite before it is developing. This specific project has been in collaboration with our peers of Microscopy Australia, another NCRIS that you will hear about. And also, despite most of Phenomics Australia support is into biomedical research, we also support environment protection and biocontrol. For example, we have projects to genetically biocontrol the mouse plagues and also to prevent mastitis in dairy cows. And we use specific drugs to control the parasitic nematodes in livestock. If you want to know a little bit more about ourselves, you can follow us on social media, LinkedIn and X, send us an email, check our website. And actually, if you want to know more about it, you can just check our latest impact report. You have your phone here, you can use the QR code and download it. Thank you very much.
Okay. Thank you, Marina. Our next speaker is Kate Miller, who is the Coordinator for Science and Partnerships for the Population Health Research Network.
Good afternoon. Thank you for having me. Really pleased to be here to speak of the work of the Population Health Research Network and also data linkage in Australia more broadly. I would like to acknowledge the traditional custodians of the land in which we're meeting today and pay my respects to the elders past, present and emerging. I'd like to start by giving a brief overview of the Population Health Research Network. So the PHRN was established in 2009. It was implemented through NCRIS, and we're a national network of several state-based data linkage units which serve each of the states and territories throughout Australia, as well as the National Data Linkage Unit, which is based out of the Australian Institute of Health and Welfare. We also have a national coordinating office which sits in Western Australia and a secure remote access data laboratory. And so the mission of the PHRN is to lead and enable the linking of data for world-class action-oriented research. So what is data linkage? For many of our health research questions, the data that we need doesn't all sit within the one data collection. And instead, we need to be able to bring together multiple data collections to get all the information that we need. But in doing so, we have to be able to identify which records in each of the various data collections belong to the same person. And so in simple terms, data linkage is a method of bringing together information derived from different sources, but relating to the same individual or event. So in terms of the types of data that we use within the Population Health Research Network, administrative data is the most common source of linkage projects. So that's data that's routinely collected as part of the delivery of a service. And so this slide here just gives you some of the examples of the state and Commonwealth level administrative data sets that are routinely linked within the Population Health Research Network. And we also do ad hoc or once-off data linkage projects using clinical registry data, survey data, and research data. And so what's linked data been used for? It's been instrumental in investigating common and rare diseases and events. It underpins a lot of our epidemiological studies in the origin, associated conditions and outcomes of disease. It allows us to compare different treatments and interventions. So if we were to link, for example, hospital data and death registry data, we can look at survival rates of 2 different surgical interventions, for example. We use it for real-world evaluation of policies and services, and also looking at safety and efficacy of vaccines. So it's been used by linking the immunisation registry with the cancer registry, for example, to look at the efficacy of the HPV vaccine in reducing cervical cancer rates. And then monitoring the health and well-being of Australians across the life course, because we do have longitudinal data over many decades for a lot of these data collections. And so linked data can also be used to support the medical product development pipeline. So it can be used in clinical trial design to support site selection and also identify at-risk populations. It can provide retrospective data on patient morbidity or service access. So for example, it can allow us to establish the journey of a participant before entering a trial to give us a idea or measure of trajectory of care. Because administrative data collections involve the whole population that access a particular service, it does allow us to look at the representativeness of a trial population, but also allows us to look at the generalizability of those trial results as well. And then further to that, it can also allow us to describe those who are not recruited or those who have not chosen not to participate. So in the US, for example, data linkage was used to measure participation rates of minority groups, and the linkage study found that there were disparities in representation, with African Americans far less likely to engage in cancer clinical trials, even though they had a far higher burden of disease than other populations. I mentioned that linkage or administrative data collections provide real-world data and also whole of population data. So it's immensely useful for surveillance and monitoring of safety and also reporting of adverse events, particularly if your primary outputs are associated with hospitalisation. For example, research has shown that administrative data is more complete than data that we can get prospectively through the clinical trial itself. And it can be the longitudinal nature of administrative data can be really useful for long-term follow-up within clinical trials, whether you're measuring health outcomes, if you're looking at assessment of long-term effects of treatment, or just looking at healthcare utilisation of patients over a projected period of time. And it's obviously a cost-effective way of doing long-term follow-up in clinical trials as well. The administrative data is already routinely linked, and so the cost associated with accessing linked administrative data is far less than collecting the data prospectively over a long period of time. And then finally, it can be used for health economic analysis through linkage of data sets such as the National Clinical Costing data collection, for example. So just to finish, I've got the PHRN website up there. I've also got a link to the metadata platform. So if anyone wants to explore the data collections that are routinely linked within the PHRN, and also a link to the online researcher training for anyone who's new to data linkage and wants to learn more about the types of data that we link, how you can go about accessing data, and also the governance that sits around data linkage in Australia. Thank you.
Fantastic. So thank you, Kate. So next speaker is Natasha Simmons from the Australian Research Data Commons.
Thanks, Bono, and hi, everyone. So I'm the Director of National Coordination at ARDC, and I'm based at the Australian Institute for Bioengineering and Nanotechnology at UQ. So great to be here. Has anyone ever had a blood transfusion here? Nobody? I had one. I nearly died, And I'm really grateful to the clinicians and to the people who helped me and to the people who donated that blood to enable me to get through that. But did you know that we also don't know when, where and how blood is collected across Australia? We actually don't have that national view. We don't track patient outcomes. No one's ever come and said to me, how are you? What happened after that transfusion? We don't track those things. We don't track them on a national level. And then, so this is just one example of one of the projects that ARDC has co-invested in to help solve some real challenges in health research and translation. I mean, another example, has anyone ever been to a hospital emergency department? Come on, everybody has, right? You collect electronic medical records, you know, when you come to an emergency department, and if you go to a different emergency department at a different hospital, they will collect different information, and that information is not connected and there's no common data model. And so you can't exchange that information very easily, and that affects patient outcomes. So we've had a project to help look at how to actually bring those emergency medical records on a common data model. Another example might be we don't have a national air health quality database. I mean, sorry, I'm finding some of the projects that we co-invest in, I find staggering that we haven't actually got so far, but we don't have a national air health quality database. We have air health information, but it's stored in different databases across the country. It might be a government database, might be a researcher database, and those things don't talk to each other. But air actually doesn't observe such boundaries, and we need to be able to run health data sets across air pollution data sets to see the effects of air quality on health. So that's another example of one of the projects we're involved in, the health and translation space. So I'll just move on to the next slide. So what is the ARDC? So of course we are funded by NCRIS, you already know that. And our mission is our purpose is to provide Australian researchers with competitive advantage through data. So we are focused on research data infrastructure, and we don't do that alone. We do that in partnership. So those projects that I mentioned, for example, the National Transfusion Data Set, that's led by a range of partner organisations like Alfred Health, the Red Cross, you know, Ambulance Victoria, all these types of organisations are involved in developing that type of national scale data assets that can benefit Australian researchers and have really good outcomes from translating that research into good patient outcomes and basically make the lives of all Australians better. So our focus is on these thematic research data commons. So here we have a data commons focused on people. So it all sounds very broad, but that's the health and medical focused data commons, which I'll explain a little bit shortly about. And we also have the planet, which is focused on environment, and HAS and Indigenous, so humanities, arts and social sciences, and an Indigenous data commons as well. So here are our audience. We have key stakeholders across government. We have people who work with researchers in universities as an example, and of course, researchers as well. So one of the main projects that we are invested in that I think is probably the most relevant to this audience is the People Research Data Commons. So you can see there, it is a large investment in a national scale data infrastructure for health research and research translation. So researchers, innovators and policymakers seek to improve health outcomes for society, but the data and the digital platforms that they need span multiple layers of government, health service operations, health research studies, institutes, facilities, and the private sector. And that's a challenge. And I've mentioned some of those examples before. So the People Research Data Commons initiative is developing, operating, and coordinating national scale capabilities to support digital health research and translation. And it is a consultative process. So co-design and co-investment are the buzzwords at ARDC. And it's what we do collectively with the community to be able to build something that is useful for research, that is useful for researchers. So what is the data commons anyway? So conceptually speaking, if you think about this virtually, it is the people, the skills, the storage, the data, the models and the compute that brings all of those things together to enable researchers to conduct world-class data intensive research. In the People Research Data Commons, or RDC for short, this is a multi-year infrastructure investment, and here are some of the focus areas for that People Research Data Commons. So academic-led health studies, so there's an acronym there, HASANDA. Some of you may have been involved in HASANDA, and I'm going to talk about that in a minute. We do have a Queensland node. Also secure research environments, advanced analytics, and Commonwealth data access as well. So the HASANDA program is a strategic partnership with the academic health research community to build digital research infrastructure, and it had an extensive consultation and engagement process to build consensus and co-design that infrastructure with the national community. What is one of the outputs of HASANDA? Well, actually, no, I'm one slide ahead of myself. Here is the HASANDA Advisory Committee. So this whole HASANDA project has been steered by some really big, important players in the clinical trial space in particular. And ARDC, for example, has a memorandum of understanding with NHMRC as well to collaborate on HASANDA and a range of other health-related initiatives. But you can see the Australia-New Zealand Clinical Trials Registry and the Population Health Research Network and a number of others. So if you're from there, thank you for your contribution. So far, it has been a very brilliant project with 72 organisations across Australia. And here's a bit of a spread. So you have 10 medical research institutes in there, 16 clinical trials networks. What did they produce? Well, here's the first thing that's come out of HASANDA. So it's called Health Data Australia, and it's an online portal for discovering health data for health research. So its main focus has been clinical trials data. So the whole HASANDA program has been focused on creating a mechanism for people to find and share and access clinical trials data, which is, of course, really an important and tricky problem to solve because there's a lot of sensitivities around that data. So the secondary aspect of, so this is publicly available now. Anyone can access this portal. It is contributed to by a number of the HASANDA nodes and HASANDA organisations. So they own, they manage the data and they manage access to the data. So when you find the data in Health Data Australia, you click on and you want to see more of it, you can click on a request this data and it will send a request to the right people who can then access your request and give you access to the data. So I think it's really a fantastic piece of work, and we're always improving this as well and building on that success. And we're now moving on to academic-led health studies, such as cohort studies as well, to be able to include in there. So here are some health-related ARDC co-investment projects that have either completed or are currently underway. I've already called out a number of those, like the Integrated National Air Pollution and Health Data Asset. There's a whole lot of examples there for you to explore. So how can you engage with the ARDC? So if you go to our homepage, you'll find out more. We've got some brochures at the front for anyone who's here on our People Research Data Commons, and you can sign up to our newsletter to find out a lot more about that. So thank you for your time. I hope you engage with us through one of our many projects and programs. Thanks.
Okay, thank you, Natasha. Our next speaker is Wojciech Kaczynski, the CEO of the National Imaging Facility.
Thank you so much, Paul. So my name is Wojciech Kaczynski, CEO of National Imaging Facility, and very grateful to be presenting here today. And just also wanted to acknowledge that, of course, National Imaging Facility is actually headquartered here in the building at upstairs. We're on level 7. So, a warm invitation to please talk to us. So National Imaging Facility is Australia's advanced imaging network. So we provide access to the biomedical imaging capabilities that researchers need to make significant impacts on major health challenges. We provide access to around about 100 advanced imaging instruments across 14 partners. And we co-employ a number of health experts, around 50 to 60 across Australia, who are dedicated to helping researchers create their imaging experiment, to set up their imaging protocol, to analyze their imaging data and so forth. We're organised into a programmatic structure. Our Advanced Human Imaging program provides access to around about 35 of Australia's highest end human imaging instruments. And they're distributed across universities and also in clinical environments to image some of the sicker subjects. We also increasingly are distributing our infrastructure so that we can provide representative data to the researchers who use us, to ensure that researchers can access a wide range of human subjects when they're doing their studies, and they can easily refer patients to our facilities as well for their studies. Preclinical and frontier imaging, preclinical of small animals and large animals. We have dedicated large animal facilities across Australia, but also new imaging infrastructure and imaging infrastructure tailored to niche applications such as plant imaging. Our new program, Imaging Data Collections and Partnerships, we are a huge data producer across Australia, and imaging data is highly dimensional and also notoriously hard to analyze and to manage. And so our new Imaging Data Collections program, which I'll show you a little snapshot of, is dedicated to helping our users use imaging data, the data that we generate, but also apply new generation techniques such as AI, deep learning. Radiopharmaceuticals are a product used in medical imaging. They're also quite an important area of innovation as well, particularly in the theranostics space. We coordinate radiopharmaceuticals for imaging across Australia, and we also help develop novel tracers to allow researchers to ask very specific questions and visualize processes within animals and the human body as well. We're distributed across 14 partners in Australia. Don't be misled by just the 2 entries there in Brisbane. We actually have a very, very large collection of instrumentation across the Centre of Advanced Imaging at University of Queensland and Hurston Imaging Research Facility. Queensland is actually one of our largest footprint of capability in Australia. And we provide a full suite of capability here, from radiopharmaceutical development through to preclinical, large animal and human. I'm going to just give you an overview of some of the specific instruments that we provide and some of the things that researchers are doing with them. So we run Australia's only 2 7T MRIs. These are the highest grade MRs available open access in Australia. And the advantage of using these instruments, which are very sophisticated pieces of kit and very difficult to use, the advantages are significantly increased signal and improved contrast. Recently, you might have seen this story in the news. It was a collaboration between Griffith and University of Queensland, where they used one of our 7T instruments to identify a structural link in the brain between chronic fatigue syndrome sufferers and long COVID sufferers, forming a very strong connection between those 2 disorders, which has significant effects on long-term treatment, perhaps. Across Australia, we run a national network of point-of-care MRs. So low-field magnetic resonance is a real area of innovation right now, and there's a number of products that are becoming available that are somewhat portable. These things look portable, but they actually weigh about 500 kilos. Emergency departments across Australia are considering deploying them so that they can move the magnetic resonance equipment to the patient rather than the patient to the MR suite. And we're helping to actually help emergency departments understand whether that's a good strategy within their context. So we're working with a number of emergency departments on deploying these and then helping to assess them. We run Australia's only open access total body PET facility. This is a piece of infrastructure for imaging the whole human body using molecular imaging. And it has significant advantages in sensitivity. So this is an instrument that is substantially more sensitive to imaging than a regular PET that you will get if you require one. What it means is that you're able to actually do things like measure total body dynamics, actually see a tracer be absorbed in the body, and you can see the kinetics of that tracer as well. So the way that the organs take up that tracer and the interaction that actually happens with that tracer. This platform is deployed at University of Sydney and is an amazing piece of infrastructure to understand the effect of novel cancer therapies. And in fact, very recently, Professor Richard Scolyer, who's one of the 2 recipients of Australian of the Year, and you may know, is undergoing treatment, an experimental treatment for brain cancer, actually had his scan done at this facility. Again, it is an amazing facility for understanding the effect of a novel treatment. Increasingly, researchers are coming to us and asking for access to ever larger data collections. And we are a big data producer, but the power of imaging really is when you can collect a large data set. And we're helping to accommodate that within our network. We have a new activity that we're deploying as part of our data program. And the purpose of that activity is to provide researchers with better access to a suite of capability across Australia. So really to be able to access all of our nodes through a single front door and to ensure that the data that is collected is harmonized across multiple sites. So this is our very first pilot. It's a collaboration across the traumatic brain community, and we're collecting a large data collection referred from emergency departments of subjects who have mild traumatic brain injury, with the intent of publishing in the long run an open data collection specifically for mild traumatic brain injury, making that available to the research community for data processing and for analysis. Partners such as AFL, ARDC are involved, and so forth. But this is something that if you are generating larger and larger data collections, this is a program that we want to talk to you about, because we want to tailor it to make it fit for purpose. Just last contact details. Our community, our user community, our stakeholder community is really important to us. So we do encourage you to please join our newsletters, find us on LinkedIn and so forth, and please do come and contact us and talk to us if you do require imaging services. Thank you.
Thank you, Wojtek. And our final speaker for today is Roger Webb, Director of CMM, the Queensland node of Microscopy Australia.
Yeah, thank you very much for having me here. You have to get used to my Swiss English, which I call Swinglish. I have a beautiful topic, Microscopy Australia. You're probably all aware of magnifying glasses, looking at things to start to explore the world. That's what kids do. We have now kits in our labs, which are 80 millions of value, and have 8 to 16 different detectors on it. So very complicated devices. It's not something I can just give somebody to use. So what is Microscopy Australia? The NCRIS capabilities is the top edge of electron microscopy, mass spec imaging, and other imaging tools like X-ray imaging tools across the country, which you can access. They all have underpinned laboratories which have basic research instruments, which you also get access to once you get access. You see, we are a national network from Western Australia, Adelaide, Flinders Uni, South Australia, Monash, ANU, Sydney, UNSW, and UQ. So a quite broad group of people working together. what do we actually offer access to? Not only the instrument itself, but also the knowledge about how to prepare samples, especially if you think a biological sample. If you take a biopsy, it's not something you can just squeeze in an electron microscope and get a tomogram of the cellular structure. You have to have the skills and the right equipment available. So we offer specimen preparation training. We also help. We offer fee for services if you don't want to send somebody, you don't have the capacity. And then we do all the types of microscopy, which we have here on the slide. And some are more centralized and others are more across the country. So for example, mass spec imaging, where you look at small molecules, is more happening at UQ and down in Adelaide. Then on the other side, X-ray tomography on material science, as you stress materials, it could be a bone density map, which you do on the stress tension, can be done nearly at all the other sites. So if you want to connect to us, please contact one of our sites. We heavily engage, and we don't produce structures, but we heavily engage with industry. And you can see here, roughly across Microscopy Australia, 150 direct industry clients every year we serve, and many others in linkage projects. Many from the resource, food, beverage area, but also in the medical devices. And these are just the contacts we had over the last 3 years at CMM as part of the local network. So I give you 2 case studies, basically, where Optima is one of the company which tries to make small detectors, which you can wear, personal devices to monitor your health. And one is about dehydration. These devices have been fabricated at the ENFF, and we do quality control. So we image the structures, we after usage, we on skin, and we do also good manufacturing documentation for them. Similar like we did it for Vaxis, which is a vaccine delivery system, which has no syringes, typically just small needles. They use our instruments for good manufacturing protocoling. So we not only explore, but you also can document and do quality control for these companies. Another example, which is now becoming a very cross-country available technology, is cryo-electron microscopy. You see one instrument here. This is one of the instruments which I told you before, multi-million instrument, different detectors, produces huge amount of data. The idea is that we have viruses, vaccine, proteins, and we try to overnight collect 100,000 individual images. And from these individual images, we reconstruct the atomic structure of these viruses and proteins. So in this case study, basically a vaccine platform has been developed between UQ and Monash University, which now can tackle down rare viruses infections in mammalians, but also in humans. And the other one, which I mentioned in the beginning, or Bruno also mentioned, was the clamp technology, which came out of the UQ vaccine development for COVID. The clamp has now become a whole new technology for holding proteins in a 3-dimensional arrangement, as we call that quaternary structure, so that we can actually give it to a patient and they produce antibody against these proteins. This is a company now hosted in UK that came out of Paul Young's development with Keith Campbell and Daniel Watterson at UQ. So we can actually image these proteins, we can image the viruses, but we can also, if we do single particle reconstruction, get to the atomic scale and help you to fine-tune things. People would like to understand where drugs are docking to these proteins. So in principle, you have access to 250 instruments plus, because we have also feeder structures at different universities, roughly 130 experts. At UQ, you're actually pitching to brains of 450 years of electron microscopy and mass spec imaging technology experiences. So you don't have to learn it, but we learn and train people. At UQ, we trained last year 750 users. They get access and they can use it as a fee for service, or they can actually book just beam time and be independent completely. We have 150 industry clients roughly across the ring and around 250,000 beam times. Sounds extreme lot, but if you want to investigate one single protein to the atomic structure, you need a few hundred hours to get there, and then also a few days and weeks for image processing, basically. So this is maybe not a big figure, but in overall, 1,400 publications are our key KPI factors. And we produce beautiful images, as you can see on the left side, nuclei just stained in tissue. So if you're interested to understand how your surface of the skin or even your tissue inside is organized, how the cells are behaving together, how proteins are arranged, or how subcellular systems are organized, we are the people to come and talk to. Thank you for attention.